Ok. Ti devo dare il microfono?
So welcome everybody, and this afternoon we have the pleasure to have with us Mattia Pelizzola, who did his PhD in biology of complex systems here in Torino, and then he did 2 postdocs in the US, at Yale and at Salk Institute. And then he moved back to Italy, where now he's a team leader at Istituto Italiano di Tecnologia in Milano and working in epigenetics. And this afternoon he will give us a talk on his research.
Pointer? Okay, I guess I should start. Okay, thanks a lot for the invitation. Thanks to Michele, to Andrea, and to everybody. It's always really nice to be back in Turin. I don't know where to put myself. Can you all see? No, you cannot. I'm sorry. I have to block the view. I will walk, so. The title of my talk is The Epigenome, a new code superimposed on the DNA. There is going to be a quite long introduction on the field for the people that are not specialists on the terminology and on the different notions. While at the end, real data, actual data, some of them actually unpublished, will be presented on the topic. Yes? Oh, I see. Okay. Can you all hear me? Enough? Okay. So I will start with an analogy. So we'll compare a cell to a town, and the nucleus and where the genome is maintained is enclosed to a library. So I'm going to make this analogy. Hopefully that this will make things more clear for those of you which are not familiar with the basic concepts in this field. So you have a town, you have a cell here, a very complex structure, and the center of the town there are typically very important buildings such as a library, which in my analogy compared to the nucleus and to the embedded chromatin. The chromatin is nothing else like the genome, the DNA, with a specific three-dimensional structure, folded. Thanks a lot. Oh, nice, thanks. And then within the library, there are many books, and the books, in this case, are going to be the genes. So the pieces within the DNA, which are actually the fundamental unit of information. Typically, you want to look for a specific gene, for a specific book. So there are going to be parts of this library which are very nicely accessible, very ordered. It's very clear what is there, how you can access it. Sometimes there are different copies of the same book on the same subject, maybe with slightly different variations. Other parts of the library are going to be very difficult, even physically, to access, or very chaotic. It's going to be quite hard to access and to read a specific book that you might be interested in. At the end, you might be lucky enough and able to find the book that has the content that you are interested in. And the analogy here is with the structure of the chromatin. Now, this looks very complicated, but we will go through this image in the next slide. But the main idea here is that there are going to be parts of the chromatin. The blue rope is basically the DNA. Parts of the chromatin are going to be very accessible, open up, and easy to access. And the gene, you can interact with the gene very easily. Other parts of the chromatin are going to be folded very closely. This is called heterochromatin compared to euchromatin. Are going to be very poorly accessible, and it's going to be difficult to reach the gene that you are interested in, so you cannot really exploit the information that is encoded in that gene. Once you have been able to identify the book that you are interested in, the book is very precious, so you cannot take it with you outside from the library, but you can make copies of it. So you go there and you make a copy of the whole book or a part of it, and what you can take with you are these copies that represents like the instruction manuals to build something, to make some physical object that is the final product, that is the fundamental thing, right? So like for example a machine. And the analogy here is starting from the DNA, which is the library. There are specific genes, which are the books. You can transcribe them, getting another nucleic acid that is called RNA, which is the copy of the gene that you are interested in. And this can be translated into the real thing, which is the protein, that is a three-dimensional physical structure that can have a specific function that is the final results, which is very important. Is a three-dimensional structure that results from the folding of a linear sequence of amino acids. I'm sure you're all already quite familiar with all this terminology in other senses. At the end, keep going with the analogy of the towns. We have different towns on the planet. Now, people in Venice and people in New York basically share the same knowledge. They all know how to build skyscrapers, but in Venice they don't have them. Also in New York they know about gondolas, but they don't have them. Each one of the city have the same DNA, but they use different portion of it. They express different parts of it, the part that is needed for them. So in Venice you're going to find things that you don't find in New York, and vice versa. This is a first clue to understand how it is possible that in complex organisms like ourselves, or in general mammals, you have the same DNA shared among all the cells in our body, but eventually the cells are able to become very different things, starting from the hair to the liver to the skin. They all share the same genetic information, but they are able to use it in a differential way, so to become something different eventually, the different tissues and organs. How is this possible? The epigenetics is the key ingredient here that allows all this very complicated process. This is the outline of the presentation. First, a panoramic on the main actors in the field of epigenetic, the so-called epigenetic marks. Some hints about their functional role, so what is the real function that they exploit, and then how it is possible to decipher the interplay between the epigenetic marks, and finally, what is the effect on the transcriptional regulation. If you remember, is the copy of the books, is the action of copying the book, which is the part that is critical to define which gene are actually active. And eventually, a hint on the epitranscriptome, which now might seem a word that is obscure, but is the same thing at the level of DNA projected at the level of the copies, at the level of the transcript of the RNA. Very simple, but nice definition of epigenetics is coming actually from the Wikipedia. It's the study of heritable changes in gene expression caused by mechanisms other than changes in the underlying DNA sequence. So DNA is always there, is always the same. But something is changing on the top of it. In fact, this part epi means, from the Greek, something on the top of, in this case, genetics, so the genome. Epigenetics, epigenome, the genome, all these omics at the end in the biology world just means something like done at the genome-wide level, not for a specific gene. So it's a way of generalizing concepts, and it also refers to the methodological and technical approach that measure things at the level of the entire genome, at the level of the entire cell, compared to the old-fashioned way of working in biology, where you were focusing on a specific gene, which is a specific book, a specific subset of the genome. So the first actor is the methylation of DNA. So we talk about the DNA, a series of letters, 4 bases and different combinations, as I'm sure you know. We are talking about 3 billion of these letters in the human genome, and we have 2 complementary strands with a given code of complementarity between the bases. One of these bases is the cytosine, here as a C. The cytosine can occur in the regular, unmodified form, or through the action of this enzyme, DNA methyltransferase, that can take a methyl group, that is 1 atom of carbon and 3 of hydrogen, from a donor, can transfer this group to the cytosines, originating the so-called 5-methyl cytosine. So eventually, the DNA methylation is nothing but a covalent modification of the cytosine on the DNA itself, on the top of the ovarial cytosine. Some of the cytosines are not going to be modified, others are going to be modified. So we say these are methylated and these are not, of course. Then, as I anticipated before, the DNA is maintained, is very long. I think it should be like 1.5 meters long if you could extend it out from each individual cell, and we have billions of cells in our body. It is maintained very compact in the chromatin, and specifically is folded in a way such as like a yo-yo, you know, around these proteins, which are called histones. There are at least 4 of them, plus variants. So that is a first level of compactness, and then it also folds up progressively in other ways so that you can really squeeze it in a small volume. These proteins, the histones, are very important units in the organization of the genome, and the tails of these proteins, so the last part of the amino acid chain, can actually be modified analogously to what I just shown for the DNA through the addition of chemical groups that can be methyl groups, even for the histones, as exactly like for the DNA, but also other groups like acetyl groups and other of those. This is what we refer to when we talk about histone marks. Specifically, the right terminology is post-translational histone modifications. As I mentioned, there are at least 4 histones. Each histone, this is the primary sequence with all the letters, each letter for one amino acid. And different amino acids within the primary sequence of each histone can be modified by the addition of this chemical group. There are going to be methyl groups, phosphoacetylation, and others. So, as you can see, the possibility of combinatorial labeling, of alternative labeling of the same histone is very high, potentially. Not all this combinatorial potential is actually exploited in the real epigenome, but the complexity of the system is remarkable. In fact, an histone code, and together with the DNA methylation, an epigenetic code has been postulated, and part of it has been clarified and verified, analogously to the genetic code of the DNA bases. Other fundamental concepts that are in common between all epigenetic marks is that they are dynamic and they are inherited. In which sense they are dynamic? The DNA is a static information. You have the 4 bases in a given sequence in all the cells of your body. Basically, it's always the same information. It's a static thing. You were born with that. You will keep it with you, basically untouched, apart from mutations or relatively infrequent events. The epigenome is much more dynamic. In fact, there are specific enzymes, which are the proteins, that are called writers because they are able to put those marks on, for example, the histone tails or on the DNA. There are erasers that can get rid of those marks, and there are readers that are other molecules that can specifically recognize one of these marks attached to it. The combination of all these 3 mechanisms, there might be multiple writers and erasers. The combination of these mechanisms is what defines a specific epigenetic state in a given moment of the life of each cell. And then is inherited. How is that possible? Is inherited first at the level of one cell that is giving origin to the daughter cell, so the cell replication. Is also inherited at the level of, let's say, transgenerationally, like to my son and so on. No, I will show an example of inheritable information from the father, for example. This is what you are suggesting, yeah. How is that physically done? It's relatively simple. So this is the methylation of the DNA. These dots are the methyl group attached to the cytosines on both strands of the DNA, the forward and the reverse strand. When the DNA opens up for the replication of the DNA, basically a new copy of the DNA is created on the opposite strand based on the original template. At this point, the new cytosines will not be methylated at the beginning just because it's a new copy that is just been added. But there is an enzyme that is smart enough to recognize that this piece of DNA is amimethylated, is partially methylated, and this enzyme will go there and will complete the uncompleted methylation status, so propagating this information and copying the same epigenetic status on the new strand of DNA. Analogously, something very similar is done at the level of the histone marks. Now, a first hint on the final results of all this set of things. The combination of the different states of methylation of the DNA and the combination of the different marks at the level of the histones, which are dozens, as I shown to you before, is basically defining which part of the genome are actually available to function in that specific moment, compared to other parts which, as I suggested before, they are basically even physically closed up, and basically they are inactive. So first hint is about identifying the part of the genome that is active compared to the one that is inactive. This is basically the, okay. But this was just a very naive explanation on what is the actual effect of the epigenetic marks. So let's go more deep into the functional role of these marks. So this is the hierarchy of things that we were talking about, the DNA that can be methylated, then fold up around histones like that, can fold up at hierarchical levels. At the end, you can even define the entire chromosome, right? And this is not a complete list of processes, a very important process in which these marks are having a strong impact. Many of these are not going to be clear to you probably, so we will go which one slide for each one of these things to try to clarify them. Let's start with the regulation of tissue-specific transcriptional programs. Is what we were talking about before. So how it is possible that from the same DNA we originate different phenotypes. It's all about using different portions of the same genetic information of the same DNA. So basically through the epigenome we can define the parts which are actually needed in that moment, originating the blood cell compared to the heart, for example, just taking advantage of different parts of the genome in combination. Cell differentiation. Cell differentiation is exactly the process that, starting from the embryo, at the end will originate the whole organism. Everything basically starts from the stem cells. The pluripotent stem cells are able to be differentiated, are able to transform themselves in all the cell types, in all the tissues and organs which we have in our organisms. This is typically depicted through this surface that was drawn by Weddington. I don't remember how many years ago, the 60s maybe. And basically it's like there are channels that in a more or less definite way bring the original cell to a differentiated state, and it's a relatively an irreversible process. So once the liver is formed, now it's not that the cells from the liver can revert and go back to the original stem cell. At least it's not supposed to happen. Two very important concepts that can be added on the top of this scheme are the de-differentiation, which actually is the process of coming back, and the process of transdifferentiation. Actually, these concepts are quite important in the field of translational medicine. This is the one that originates, maybe you have heard about induced pluripotent stem cells, where you start from a cell that is differentiated. For example, you take a cell from the skin of your body, And through the action of a specific cocktail of transcription factor, a cocktail of molecules, this cell is convinced to dedifferentiate. So the cell has the potential to come back to the original primitive state of more or less pluripotent cell. Just this path is typically not used in an healthy body, but it can be a very important tool in medicine. Why? Because, for example, if you have a disease with a specific genetic mutation, you take the cell out from the skin. With that cell, we'll have that specific mutation, but maybe the skin doesn't really care about that mutation that is causing problem at the level of the brain. So in the lab, you can actually now recreate the cell from the brain, starting from the skin cell, exactly through this process, and then going down to the level that is interesting to you, for example, the cell from the brain. So you have a very incredibly powerful tool to manipulate the differentiation stage of your cells. And it is also possible to transdifferentiate going directly to one differentiation state to another one without passing to the original pluripotent state. Other fundamental way of action of the epigenome is through the modulation of the binding of proteins. So the proteins are these physical three-dimensional things which are interacting with the genome itself and are fundamental in exploiting the information encoded in the genome. So we say that the proteins really bind the genome. The binding of this protein can be strongly inhibited many times if the DNA is methylated in a given specific region. So this is a symbol that we use to say that this inhibit, in this case, the binding of this protein. While if the same location is not methylated, then the protein can bind. There are going to be other factors, other proteins, which actually don't care about the methylation status of the genome, or even the opposite, which are attracted by portions of the genome that are methylated. About the histone marks, two different hints on how the histone marks are influencing the binding of protein on the DNA. First, depositing acetylation marks through the charge of these chemical groups opens physically up the DNA, making it much more accessible to the arrival of regulatory proteins. So these are really a physical action. On the other hand, histone marks can be seen as docking stations, which can attract proteins which really like this mark and are very complementary to that mark. So it's a different concept, right? And it can be much more subtly modulated. Another important evidence is the action of the environment and of diet on the epigenome. So the epigenome is not static by itself, not only, but is very much influenced by our interaction with the environment, in the sense of what we eat, what we breathe, and where we live in and what we feel. For example, the effect of stress. Right now I'm a bit stressed, so my epigenome is changing. If in your early life you had a very easy environment in which your mother was very careful and close to you, that is very important to modify the epigenome in the hippocampus in your brain in terms of methylation, for example, of the promoter of this specific gene, NR3C1. If that promoter is not methylated, then it can be bound by this protein. And the binding of this protein allows the expression, the activation, the usage of this gene. If the environment was very hostile, and this of course has been studied in mice, the same gene is going to be methylated. If it's methylated, it cannot be bound by this protein. If this protein is not bound, the gene will not be used. So this gene is going to be silent, inactive. An RNA, a copy of the gene, will not be created, so you will have a remarkable different expression of this target gene, which is critical in determining how you're going to react to the stress. And this responsiveness will be maintained during your growth. So even the adulthood, you will be affected by an epigenetic, impact of the environment that you are inheriting from cells that you had when you were a newborn. The epigenome depends very much on what you eat, even just because the methyl groups themselves are coming from the food. So there are specific species of strains of mice that whose the color of their skin can change accordingly to the methylation of a given gene, which is going to be very much influenced by their diet. So giving them different food will make really a different, a major effect, visible effect on these mice. In our case, the analogy is going to be about what we breathe, for example, if we smoke or not, and the diets that we have. All these effects can be transmitted and the second generation, for example, the fetus, or even the third generation, so the reproductive cells of the future children. It's not just from the mother, it can also be transmitted by father. For example, it has been shown, again in mice, that if the father has a diet that is very rich in fat, his daughters, and not his, so only the female offsprings of this father will be more predisposed to have diabetes. This is mostly because of the demethylation status of one gene that is available on the X chromosome that will be transmitted from the father to the daughter. On one hand, this is quite scaring, right? But on the other hand, this actually is very powerful and provides a lot of opportunities to study those things and to actually intervene on these things. Silencing transposable elements. There are pieces of the DNA which are called transposable elements that typically are silenced, are blocked because they are dangerous. They are blocked exactly because they are methylated. So other genes can function properly. While when these transposable elements are not methylated anymore, for example, through the action of stress on the epigenome demethylating these regions, these transposable elements can be activated, can be expressed, and can literally jump in other portions of the genome. Sometimes they can even conflict with the actual insert at the level of a given gene, disrupting their activity. So it's a very important control mechanism that is done from the demethylation in this case. When something goes wrong, epigenetic marks can be associated, unfortunately, with diseases. So this is a very preliminary list of the mark, the specific alteration, the consequence at the level of the molecular biology, and the actual disease and prominent genes which are linked to the epigenetic modification for each case. A hint about this process is we have to come back a minute to this landscape, the Weddington landscape. In the original picture, there were actually genes below it that were with ropes, like they were able to pull into the form and to give a specific shape to this surface. But this is a static representation of it. The real representation should also take into account that while the cell is rolling down into a differentiation and the epigenome is changing, is modulating, so the surface will actually will change during this process. So the epigenome is a fundamental information that will direct into different directions, but will also influence the course of this path. And what's going to happen is that the epigenome in your disease cells is going to be quite much disordered compared to a normal tissue, where a given level of mark, in this case the methylation, 80% methylation, for example, on a scale from fully methylated, fully unmethylated, is going to be very much disordered during the development, in this case, of a carcinoma. So the methylation level is not anymore confined within a given range, but is free to move within a very large range. This will originate the fact that now you might have cancer stem cells opposite to the health stem cells. These cancer stem cells are much more likely to differentiate in the real cancer cells, which are like the differentiation endpoints, but from the point of view of the disease, which are going to be much more closer to the pluripotent state compared to the real differentiated state. So all these states are very unstable. You see that the valleys are not very deep. It's a very chaotic region, so you can move very fast from one state or go back to the opposite. Till one specific cell will find a set of epigenetic marks that will give her some competitive advantage. That cells might be very fit in that environment, and that will originate the cancer, because that will prevail, will be the clone that will originate actually the tumor. In the reality, how you can measure is basically related to this. So it's related to the fact that while the cells are differentiating, parts of the regions will be silenced, will be closed up, will not be in the liver, that part will not be used anymore. But in the cancer stem cells, that part that is highly methylated, highly compact, will be demethylated, will open up, will be more accessible. So in practice, you will have both variance and, in the case of methylation, lower frequency of high methylation events, which is critical to make that parts of the genome available again that will totally change the phenotype of the cell, that will not be a liver cell anymore, but can be something different at this point. Okay, we are talking about dozens of layers that can interact simultaneously, and so it's obvious that we are very much interested in the interplay between them, right? And it's going to be quite complicated to understand their interplay. So we are dealing with a very complicated machine. This is from Ruby Goldberg's. It's a very complicated machine to do something simple. At the beginning, we really don't understand this complexity. So the first thing that we typically want to do is to map the individual component to try to understand what are the pieces of this machine. Which, by the way, is a napkin that will clean your face while you are moving the spoon eating. The second thing that you might want to understand, okay, I see there is this component that we call it blade, but we don't really know what a blade is doing. So to understand that, it might be easier to check into other examples in which the same object is present. We might start understanding about the function of that component. That will also give a clue on the overall machine. Third thing, we might try to interfere with the machine itself. We might replace the blade with the monkey, and we can see what happens. Again, we can learn something. The analogy here is that the mapping of individual components is the genome-wide profiling of the epigenetic marks. Studying several cases where the component is present means taking advantage of large-scale public databases where dozens, hundreds of thousands of experiments were already deposited there. And perturbing the system can be physically done through the epigenome editing technique. So let's go through these points one by one. The main technique to profile the epigenetic marks are this called sequencing techniques at this point. The RNA-seq is the one that allows you to measure which genes are transcribed, which genes are used, because you select the copies that you made, you remember, from the book. You isolate that portions of those molecules of RNA which are in the cell, and you can actually physically take them and sequence them in a machine that will provide you the exact sequence of base in that set of fragments. You can do something very similar for the histone marks and for the methylation of the DNA. How is that done? For the histone marks, you're going to have the tails, which are the chemical groups, or the same technique can be used to profile where the proteins are interacting with the DNA. It's called CHIP. It means chromatin immunoprecipitation. This is the chromatin. The immunoprecipitation means that you're using antibodies, which are molecules that are specific to your target. For example, I want to know where the green ball is, I give an histone mark, or where this protein is. I use different antibodies. I can purify this. You see, you cut the chromatin in pieces and you only take the pieces that have the specific target of your antibody. And then you are able to take this and throw away all the rest. At this point, through a chemical reaction, you can isolate the piece of DNA that is attached to these regions so that you will be able to sequence only this and discard all the rest. So you will know where the green stuff is and where this protein is. For the DNA methylation, is slightly different. The critical point here is that some cytosines are going to be methylated, others not. If you use this molecule, the bisulfite, if you treat the DNA with this molecule, this bisulfite works in a way that the cytosines that are going to be methylated will be protected by the action of this chemical, while the other cytosines will be transformed into another base. So now you're going to sequence those sequences, and where you will find the C compared to the original reference sequence of the human genome in this case, you know that this cytosine was methylated in your samples, while the other cytosines, you will read them as a T, as a thymidine, as another base. In practice, you don't really sequence the whole fragment, you sequence the extremities of it, just a detail. And at this point, the machine will throw up to you all the sequences that can be 50, 100, 200 base pair long. And the point is to understand from which part of the genome these sequences are coming through a procedure called alignment to the reference genome sequence. The hits, the actual important data from your chip experiment or from your DNA methylation experiments are going to be something like this on the left or on the right. On the left for the chip-seq, so where the protein is interacting, you're going to purify all the fragments of DNA from that region. Specifically, you're going to have the sequence of all these parts, either on the left or on the right of your fragments. Altogether, this creates these two distributions that jointly can derive you this distribution that will be a proxy of the points of interaction of the protein. For DNA, it's relatively similar. So you have reads containing the methyl cytosines. Other reads will not have the methyl cytosines. At the end, this is typically represented like a bar that is a quantitative signal from 0 to 1, 100% methylated, meaning all the cases where I've measured the methylation for that cytosine was methylated, so you have 100% methylation. Like, for example, here, only 1 out of 4 cytosines was methylated, also because you don't work at the single cell level very often. You have a population of cells, so the signal can be slightly different from the different cells. So you have percentages of signal, fully methylation or partial methylation. This is typically a base resolution measure, so one cytosine, one base in the genome, you know if it's going to be methylated or not. This is a low resolution. Excuse me? Of a population of cells. Not the whole tissue, much smaller piece of a tissue, but yeah, conceptually yes. Or it can be a cell line, which is not a tissue, is a purified cell with a given characteristic maintained in the lab. Okay. While for the chip-seq is a more low-resolution technology, you will only have a broader region, maybe 200-300 base pair long, so letters, which will be a proxy of the physical real point of interaction of your protein. Eventually, you're going to compare the signal that you have on the genome with another experiment, which is the same experiment without using the antibody that will represent your background signal, and you are going to look for the regions of enrichment of this signal compared to the background. Relatively easy. I mean, relatively straightforward for you, I'm sure. Just another important hint, some of the signals are going to be very, very sharp, very well defined, very in a particular region. Still low resolution, but well confined within a region. Other signals are going to be huge compared to the genes, compared to the transcriptional units, might even cover the whole gene or entire regions spanning many genes, many megabases, and the signal might be very chaotic and not maybe sometimes so much meaningful in terms of shape within these huge regions where we are most interested about this region is tendentially positive for this mark, while this region is tendentially negative. So it's a more qualitative interpretation in this case. Oh, I think I have a problem with the bathrooms. There is a... Spero che ci arrivi. 1%. Just okay. Sorry about that. Okay, I think in order to save time, I will skip this part. It's about taking advantage of public. Let's just look at this slide. In the public databases, at this point, you have data, for example, about the epigenetic status in many, many different tissues, healthy tissues or tumor conditions within the same tissues. It's a very powerful information that is critical to understand how the different marks are related to each other. It's a very convenient information to use. It's quite tricky, it's difficult because it's coming from different labs, different techniques, so the normalization or the processing of the data is absolutely critical. But once you cleaned up in an acceptable way, you have a very terrific information that is that provides information about the functioning of your system within many, many different contexts. That is typically is the limiting point, right? It's a very complex system. You don't have enough cases in which you have information on the same system. While at this point, from public databases, there is a good amount of information about that. Okay, last point here was we can actually interfere with the machine, trying to understand how the different pieces are attached to each other. You can do that through the Epigenome editing techniques. Just a hint about that. For example, this is a region that is highly methylated, a lot of black dots, so this gene is not actually functioning, is inactive. In fact, there is also this repressive histone mark there. Through the action of these enzymes that can be directed physically and specifically to this region and not in the rest of the genome, it is nowadays possible to demethylate that region and so to express the downstream gene. And again, the key point is that you're going to do that specifically within only one region, the region that maybe is very critical for the disease. While right now, as a cure of cancer, they already are using drugs which interfere with the epigenome, but these drugs are relatively primitive in the sense that, for example, they demethylated the entire genome. Not fully, but they have a very strong demethylating action, which is not specific for a given portion of the genome. Still, they are helping. In fact, they use it. They've been approved to be used. But you can understand it's a very primitive logic, while now we have very powerful methods to go directly on the spot that is critical. Vice versa here, you can interfere with the histone marks, which are typical of this region. And this region is called an answer because in the 3D structure of the genome is very far from this gene, but in the 3D structure, sorry, in the linear structure of the DNA can be very far from this gene, but in the 3D space it can be very close, it can be fundamental. This region can be critical for the activation of this gene. And this interaction is really due to the presence of these marks here. If you interfere with these marks, now they are gone, now all this 3D interaction is gone, now the gene is not transcribed anymore. Okay. So at this point, you understood that a major effect of the epigenetic marks is at the level of the transcription of the genes. So the activation, the usage of the genes. But it is also clear that there is a very complex interplay between a multi-layer system of different marks, especially the histone marks, which have dozens of them. Even for the methylation, there are several types. It's not just so simple as I explained to you. So we talk about chromatin states. A chromatin state within the genome is a region where there is a given pattern of absence or presence of marks. In this case, only 5, but it's just a schema, okay? And it's like if you color the genome with the same color, whether you have similar patterns of marks, which are significantly present in your genome. So the trick point here is to segment the genome in the right pieces, which is not available a priori, and to see how many pieces have the same code. In practice, what does this mean? So these are going to be the genes. There are combination of marks, literally these marks, like the histone number 3, the fourth residue, the fourth amino acid, which is a K, so is a lysine. If this piece of that protein has two types, two molecules of methylation, is dimethylated, we say that it is H3K4 demethylated, and this together with these other marks define the epigenetic state of this piece of chromatin, and the characteristic of this signature is that this gene is going to be used, is going to be active, is going to be expressed. But this fingerprint, this footprint, this signature of the epigenome is typical of the left, the start part, the beginning of the gene, while within the gene there is another there is another combination of marks which means that the gene is actually physically used in that specific moment. So we have already two different kinds of signatures with two different meanings. This is for the enhancers, the things that I told you that in the 3D structure they can interact. And there are other signatures that are typical of genes which are not used. So we already have a little bit a clue on the different combination of these marks and their effect on the transcriptional regulation, whether a gene is active or is inactive. The other two main actors here that we cannot disregard are the polymerase itself. What is the polymerase? The polymerase is a very complex molecule composed of many proteins, which is the one that physically go there on the DNA and go over the whole gene, starting from the beginning, which is called promoter, going through all the gene body till the end of the gene, and it is actually transcribing the gene, is making the copy of the gene, is creating the RNA molecule that we were talking about before. What's going to happen to that RNA molecule? Here you have the gene. The RNA molecule is synthesized, so you create a first version of the RNA molecule. Is more complicated because there is a first version that is immature, is not ready yet, and then that will be matured, discarding some pieces of it, so you will have the final functional form of the transcript that is, if you remember, is the instruction manual that will originate the actual protein. But now we have to take into account that this can be degraded in your system. So there is a way to control the speed at which this molecule of RNA is degraded. At the beginning, you had the speed at which each gene is transcribed, so at which this molecule of RNA is created, and the speed of this at which this molecule of RNA is matured into the mature transcript. So the combination of all these three rates, the rates of synthesis, processing, and degradation, is the one that will define exactly how much molecule of transcript you will have available to create a given protein, which is the final important product, okay? So if you don't take into account all this underlying complexity, you're really simplifying the system, pretending that a gene is active in a given moment. And nowadays, it is possible to understand how all these steps, the progression of the polymerase within the transcriptional units, and the life cycle of a molecule of RNA, how these steps are related to the relative epigenetic control, because the epigenome is controlling a lot about these different steps. And fortunately, it is possible through mathematical modeling to get a hint on the quantification of these different steps. So we do have powerful experimental methodological techniques that allows us to study quantitatively how these layers of regulation are related to the control from the point of view of the epigenetics. It is a relatively nascent field. There is still a lot of work to do, but is opening up a very powerful details on the way the genes are actually used. Final set of like 6, 7 slides. Let's talk about another layer of epigenetic. It's not, I would not call it even epigenetic anymore, but the concept is very similar. So we were talking about the DNA, okay? The DNA can be methylated. This is the methylation of the DNA. The DNA is transcribed in a given part of where the genes are, giving birth to the RNase molecules. The RNA molecules are just other pieces of nucleic acids, which is not the DNA. Also, these pieces of these molecules can be methylated exactly like the DNA. This is the methylation of RNA. Complicating things, this time is not the cytosines that will be methylated, it's the adenine, it's a different base. But the idea is relatively similar. And again, the important thing is that this modification is going to be reversible, it's going to be dynamic on the top of something that is relatively static. Okay, the origin of this is not static, it's highly dynamic, it's the part of the DNA that is expressed. But once this is created, on the top of this, there is something that can change during the life of this RNA. So this is your transcript from the beginning to the end. This is the profile of the frequency of these methylation events, which are very often occurring at the end of the transcript. The reason of this is that in this special part of the transcript, there are a lot of important regions where that transcript is interacting with other molecules, which are critical to define where that transcript has to go within the cell, whether that transcript has to be degraded or not. As I anticipated to you, the RNA methylation is dynamic. There are writers, there are erasers that get rid of them, there are readers that can recognize them and influence their processing, their translation, so the creation of the protein, their stability, whether they are degraded or not, where they have to go within the cell, the export from the nucleus within the cytoplasm, where the protein is made. All these things are partially under control of the epitranscriptome. A given molecule of RNA can be methylated in different portions simultaneously. These are real data from our lab. So you have transcript on this on the x-axis, and is read when the methylation group is present. So there are going to be RNAs which are going to be methylated in the first part, in the intermediate part, and not at the end, and so you have many combination of signals. But what is the relationship between this combinatorial pattern of methylation and, for example, the rates of synthesis, processing, and degradation, so the life of the molecule of RNA? We are currently studying that, and the first hint is that if we take molecules of RNA that are methylated, and you compare them to molecules of RNA which are not methylated, and you compare exactly the speed at which they were synthesized, you will find out that the molecules which are methylated typically are characteristic of genes with a very high synthesis rate compared to those that are unmethylated. It might seem a paradox, but the same thing is happening at the level of the degradation. Transcripts that are methylated are going to be more highly degraded, more quickly degraded than transcripts that are not methylated. This is especially occurring when you look at the methylation in a given region of the gene. This is the first part of the transcript, not the gene, I'm sorry. And this is just the probability of the significance of the shift between these different distributions in different contexts for the three rates of synthesis, processing, and degradation. So this is about this, and this is about this. So there is a given pattern of a context-specific association between the epitranscriptome and the rates of the life of that RNA. So to make a long story short, this is the consensus after multiple analysis. So it seems like the final part of the transcript, if it's methylated, that is going to be quite important for the degradation of the transcript. The more methylation there is, the more frequently and fastly that transcript will be degraded. The same thing for the intermediate part of the transcript. But interestingly and surprisingly, the first part and the intermediate part if they are going to be methylated, they are also strongly associated with the genes that are more fastly with a higher synthesis rate. So it is sort of like if there is a feedback loop from this transcript, which is already there, is already present, is transcribed on the transcriptional regulation itself for the creation of the new molecules of RNA. And so this association between degradation and synthesis might seems to be contradictory, right? You have higher degradation and higher synthesis at the same time. Well, what does that mean? So the degradation rate is basically a proxy of the responsiveness of your system. So if the transcript is unstable, is highly degraded, that molecule is very responsive to changes, so it can change the level very fast in time. Now, if you have a given pattern of synthesis rate during time for a given gene, and you have the same identical pattern of the degradation rate, for example, they are both going down in this case, or in this case, they're both going up. That means that the degradation rate is contrasting the transcriptional activity, because a gene is more actively regulated, but is also more actively disappearing, so it's contrasting it. While if the patterns are opposite, the degradation is cooperating, is strengthening the transcriptional regulation, right? Because the gene is more actively transcribed and is also stabilized, is less degraded. So this is the first hint on how the two things can cooperate. There are even cases where the gene is not changing during time in the synthesis rate. The transcriptional regulation is steady, but the stability of the transcript is changing over time. This results in a very important modification of the level of the transcript at the end, which are not in charge of the control of the genome this time, but is just about the stability of the molecule of RNA that has been created out from the genome. So the final interpretation about all this has been the formulation of the fast track hypothesis. Basically, a transcript that is methylated as like a priority line to board into the airplane, because we are proposing that it's synthesized more quickly, is processed, is transported, is translated into the protein, and is degraded more fastly than transcripts that are not methylated, which are much more slow. And again, the degradation, let me stress that is a very important point, because if the transcript was not degraded very fast, that molecule of RNA was staying around, was keep originating potentially the protein, so the level of the protein was less dynamic. So if something is changing here, and the transcript was very stable, it's not so easy to influence the level of the protein afterward. While if also the transcript is degraded immediately afterward the creation of the protein, then immediately, if something is changing here, the signal is propagated and is originating a new level of protein, which is the final product that we really care about. And just the last two slides. Now, all these things, what I talked before, so the regulation of the chromatin through the epigenetic marks, the methylation and histone marks, and the regulation of the life of the transcript and its methylation. All these things are physically occurring in very similar parts of the genome. They are really physically close to each other. So we were wondering whether there is a physical interplay between this layer of regulation. So the first very naive thing that we did is that we took publicly available data with I think 70 different histone marks and transcription factor profile, and we tried to see whether there was some just correlation, association between the signal of the binding of these marks within the genome in specific position, and whether those specific positions were corresponding to transcripts that were methylated in the specific position. And we ranked them in this way through a simple odds ratio. And there is one of them, sorry, I'm calling it protein X because we're trying to validate that, so that's going to be secret, sorry about that. There is one of these which is strikingly associated. So this protein is going to bind the chromatin, it's not going to bind the RNA. It's going to bind the chromatin exactly in correspondence of the position where the originated transcript is going to be methylated. In fact, the co-occurrence is quite striking, that the spatial co-occurrence aligned to the position of the methylation of the RNA is remarkable correlating with the presence of that protein on the chromatin, and also for other marks that were maybe less correlated, but still quite interestingly associated. Vice versa, which is the mark that is the best predictor of the epitranscriptome? The mark on the chromatin, again, is DNA methylation. The methylation of DNA is the best thing that you can use if you want to predict blindly which part of the transcript is going to be methylated. But in the opposite way, so the lack of methylation is going to be a proxy for high level of methylation in the RNA, while very high level of methylation are going to be a good proxy for the lack of methylation in the transcript. Okay, the area under the curve, so the predictive value is quite poor, I have to admit it, but it's the top number one, and it's remarkable to think about something that is apparently totally disconnected, is about the DNA itself, buried within the chromatin, and the lack or the presence of that mark is very informative about the methylation, so the same mark, the methyl group attached not to the DNA, but to the transcript that will be originated by a given part of the DNA. And with this, I think I'm finished. I would like to thank people in my group, Stefano and Eugenia, and also Mattia, which is here and works with Michele, and Bruno Matti's lab, which are the collaborators that generated all the wet lab data, and Lara Lusa for collaboration statistics, and other collaborators. And so thanks to you for your attention. Well, I'm not expert in those now. You are catching me a bit unprepared, but the interaction with the environment is a very critical component here. So it's not just about we have a characteristic and the other cells and our children will inherit it, but the interaction with the environment. So what the giraffe will do with the leaves and the tree is going to be very important. So it's a very complex thing, and we have a relatively naive understanding of it still. But we start having now the right tools to study it. So it really started everything like, I would say, 10, 15 years ago in this scale, at this level of comprehension and accumulation of the data. So we are really starting now. Okay, I'm not a specialist in that specific parts of this field, so I cannot provide a very precise answer, but I can give two, I think, important informations. One that the epigenetic marks are not present in all the species. For example, yeast doesn't have the methylation of the DNA, but still has the histo marks, for example. Also, like C. elegans, the worm, that is a very common model system for multicellular organism, analogously doesn't have the methylation of the DNA. Bacteria, basically, they will not care about epigenetics almost. They have some components, some primitive form of it, but so the appearance of the epigenetic marks is really tightly related with the complexity of the life form. This is one thing. The second thing that's very important in respect to what you were mentioning is that so on one end you have the evolution of the information encoded in the DNA, right, which is relatively slow changing, I would say, compared to the difference, to the speed at which these marks can change. I mean, these marks are changing during our aging while we eat, and so it's a very fast phenomena. And important parts of those can be maintained and fixed and transmitted at the same time while they are highly dynamic. It's important to consider that the mutation in the DNA and the mutation at the level of the epigenome are not two independent words, but they do influence each other. So the mutation of the DNA, while it is lower, it can influence the great deal of the epigenome, and vice versa, the changes in the epigenetics can influence the DNA itself. For example, the methylation of the cytosines is a mutagenic modification of the DNA. The cytosines that are methylated are less stable than those that are not methylated and can more easily change into other bases, creating a real mutation encoded in the static sequence of the DNA that will be, again, transmitted and much more difficultly reverted. So this is another information that has to be taken into account when reasoning in terms of what you are proposing. Yeah. I hope I replied enough to. Thanks.
Giving a talk like this is very dangerous because then like questions which are not supposed
Yeah, yeah.
to use
I know, I know.
in other contexts.
No, but they were very nice questions. So.
Okay, if not, then we again.
Thank you.